A valuable way to study a gene's function and interactions is to compare the phenotype of a wild-type organism with one in which the gene's activity has either been inactivated or knocked out. In the past, knocking out a gene in an organism was very tedious, expensive, time-consuming, with few laboratories possessing the necessary resources to perform such experiments. Thanks to the development of CRISPR technology, gene knockout experiments have now become simple and accessible for almost any lab to perform. In this CRISPR-based strategy, a Cas9 nuclease generates a double-stranded break at a genomic site specified by a single guide RNA. The DNA break is then repaired via the cell's non-homologous end-joining repair mechanism, a mechanism that introduces site-specific insertions and deletions, indels, that can effectively disrupt the gene's activity. In this video, we will take you through a step-by-step case study of how CRISPR was used to develop a biallelic LIF knockout in mouse colon carcinoma cells. Our case study will also demonstrate how the knockout was verified using the Surveyor Assay, as well as Sanger and next-generation sequencing methods. First, 3 sgRNAs were designed against the mouse LIF locus. It is common practice to design more than one sgRNA to increase the likelihood of a successful genomic edit. Software analysis was performed to ensure that the sgRNAs had no predicted off-target binding sites. The selected sgRNA was then cloned into ABM's all-in-one sgRNA and Cas9 lentivector. In this lentivector, expression of the sgRNA is driven by the U6 promoter, which is a strong constitutive Pol3 promoter. To enable direct screening of the Cas9 expression via puromycin selection, the Cas9 was incorporated into the Cas9 2A puro cassette driven by an sFFV promoter. This all-in-one lentivector was then packaged into a recombinant lentivirus using ABM's second-generation lentiviral packaging system. The recombinant lentiviruses were then transduced into mouse colon carcinoma cells at a multiplicity of infection of 5. After puromycin selection, surviving cell colonies were isolated and their genomic DNA extracted. A Surveyor assay was then performed to confirm whether genomic editing of the LIF locus has taken place. In this assay, the edited DNA strand is denatured and rehybridized with the wild-type strand to create heteroduplexes that are mismatched at the mutation site. The Surveyor endonuclease cleaves at the 3' end at both ends of mismatched sites, producing cleavage products that are detectable via gel electrophoresis. In our case, the Surveyor assay produced 2 additional bands aside from the wild-type band from the colonies 3 and 6, indicating that a mismatch and consequently a successful edit had occurred. On the other hand, colony 2 did not display any additional bands, indicating that there is no successful editing. To further characterize the nature of the knockout, colony 3 and 6 were analyzed via Sanger sequencing. Sanger sequencing results showed that only one mutant sequence was detected in colony 3. This indicates that these cells are likely only heterozygotic knockouts. On the other hand, colony 6 showed 2 different mutant sequences. Further studies were performed on colony 6 to identify whether a frameshift mutation had occurred. A frameshift mutation disrupts the open reading frame, resulting in a nonsense-mediated decay of the mRNA transcript. First, colony 6 was serial diluted into 96-well plates for monoclonal selection. Genomic DNA was then extracted from these clones, PCR amplified, cloned, and sequenced. Although clones 6A, 6B, and 6D all showed biallelic editing, sequencing results only showed a biallelic frameshift mutation for clone 6A. Further sequencing of clone 6A showed that only 2 mutant alleles were present, either the 2BP or the 4BP deletion, and that no other wild-type or other mutations were detected. Sanger sequencing is a useful tool for obtaining sequencing information for up to 100 clones. However, this means a large proportion of the population is not accounted for. For this reason, next-generation sequencing methods can provide a significant advantage, as this method enables the sequencing of hundreds of thousands of alleles at one time, generating a more complete dataset. To evaluate knockout success, next-generation sequencing was performed at each phase of the project. As expected, NGS results showed only wild-type sequences before editing was performed. After Cas9 and sgRNA delivery, NGS results showed that the first round of selection resulted in a mixture of 70% edited and 30% wild-type sequences in Colony 6. This indicates that there is a mixed population of edited and unedited cells. Therefore, further monoclonal selection is required. Finally, after monoclonal selection, clone 6A showed 100% edited sequences with no more wild-type alleles remaining, indicating a fully validated and successful gene knockout. Functional studies of LIF expression levels on the resulting cell line indicated correct CRISPR targeting in the cell line. This data is courtesy of Dr. Robert Jackman from Boston University. The CRISPR-based gene knockout technique is expanding the horizons for laboratories everywhere in an unprecedented manner, enabling scientists to explore gene function and gene-protein interactions like never before. Backed by over a decade of experience in cell culture and cloning, our ABM team of scientists are equipped with the latest CRISPR tools and technologies to carry out any knockout in any cell line you desire. Our custom knockout cell line generation service offers sgRNA vector design, construction, delivery into your desired cell line, clonal screening and selection, and knockout validation by Sanger sequencing. Our CRISPR Knockout service also includes a wide array of additional services, helping you accomplish whatever project goal you have in mind. Visit our website today to learn more about how we can help you with your gene editing project.